Hi everyone, thanks for joining us today and welcome to the 11th in the Keeping It Real series. So I'm Anna Richards, Commercial Lead at Vitaccess. I'll be moderating today and I'm joined by Mark Larkin, CEO, and Sally Vincent, Senior Consultant at Vitaccess. So just give you a little bit of background on these guys. Mark has got more than 20 years of experience in consulting, specializing in European pricing and reimbursement. So when he founded Vitaccess, he sought to blend scientific expertise and technology to better represent the patient voice in drug development and market access. So you'll see a little bit more about this today as well. Sally is a senior consultant at Vitaccess. She leads the design, development and implementation of our patient-centered research projects with a focus on both qualitative and quantitative methodologies. So over the next 35 minutes, we'll be talking about capturing, characterizing and quantifying micromoments in patients' daily lives using digital real-world evidence technologies. And I'll take you over a little bit of what we do at Vitaccess, and then we're straight in with Sally to give us an overview of micromoments, and then both speakers will be talking through the challenges and solutions by showing you some case studies today, with Mark summarising the strategies for quantifying events. And we'll finish up with a Q&A session. So please feel free to put your questions into the Q&A box on the side panel. You can add them at any point during the webinar. I know we've had a few ahead of the webinar as well. So to begin, this is Vitaccess. So we're a patient-centric research organisation, and we have 5 complementary core capabilities that you can see here. And these allow us to give us a collaborative approach in conducting global digital research. And you've got from concept to implementation, we help generate these real-world data and insights. So we've got these creative patient-centric solutions. So you've got here the app or website-based data capture through to the real-time research portal for total analytics capability. And you can see here that we've got the flow from the type of participant data. So patients, clinicians, or caregivers can be reporting this data. And we can do this through site and or community recruitment. Then that is basically based on the modular architecture. So you can have studies that are 10 times faster and let you see the outputs in real time. But let's get to the juicy stuff. And I'll pass you over to the experts. I think we're starting with Sally today to tell you a bit more.
Thanks, Anna. Hi, everyone. So what I thought we would do first is just define what we mean by a micromoment. So in this context, in real world studies, what we mean by a micromoment is a change that happens sort of minute by minute in a patient's condition, and it's a burden. They're typically short and discrete, and they always occur, or they often occur, outside of the interactions with a person's clinical team, and that's why it's also really important to try and capture these. And then it's all about kind of how valuable, we want to kind of go on to talk about how valuable they are to capture these close in time micro events for real world research. So thank you. So I'm just going to talk through for the next couple of minutes about the different types of micromoments in the patient's daily lives. So firstly, if we start with the flare-up of symptoms, so these can last for hours, days, and can really vary, and therefore make it quite difficult to report back to clinical teams. So an example here is something like eczema. So eczema can appear and disappear, and it can be really quite difficult to remember when exactly this flare-up happened, for how long, how severe it was. And so this is quite an important one to try and capture. Another one is fluctuating symptoms. So symptoms can fluctuate, again, on a kind of daily basis. And an example here is something like pain. So pain you might have for hours, you might have for days, it can really vary. And again, this is quite difficult to remember when you eventually get back to your clinical appointment, to remember when exactly that pain happened and occurred. And then moving on to impact on quality of life. So a patient's quality of life can, again, the impact on their quality of life can vary. It just depends on what's happening in the moment. And it can be quite difficult to, again, report back to a clinician. And this will then also, this sort of links me back to the next point, which is daily activities. So if we're looking at daily activities, and an example of something that I can think of is sleep. So, you know, sleep can vary from night to night, and actually it's quite difficult to look back and think, well, how well did I sleep? So, I mean, if I asked you all, how well did you sleep 3 nights ago? I wonder how many of you would remember. I'm not even sure I'd remember how well I slept last night. So I can also understand how I would not necessarily be able to answer that 3 nights ago. Then moving on to work productivity. This is another one that can be difficult to remember how productive you were at work. So an example here is if we were to think of the number of hours missed, you know, in your work, you might one day miss pretty much all the day, and then it might be easier to remember that particular day because you missed most of it. But you might on another day only miss an hour, and so then it's quite difficult to remember then when and what days you spent and how many hours you therefore missed. And then lastly is treatment adherence. So if we think of like the pill counting method, for example, which is often used in real world studies, where we're kind of looking at how many missed doses participants have had, you know, how many, like they're counting how many pills they've taken. If we think of something like pain medication, then because it can vary and your symptoms of pain might fluctuate, it's then quite difficult to remember when exactly you took your pain medication. So again, it's quite an important micro moment, and it's quite an important thing to capture. Thank you. So lastly, I just want to spend some time just kind of going through what problems are solved by capturing these micro moments, and then we'll go on to talk through some actual real life kind of case studies, which will give some more kind of context here. So firstly, capturing a micro moment. So what problem does this solve? It solves the fact that we can provide more granular data on the burden of disease, which then complements the randomized control trials. And the main thing here is that it addresses so it basically, in terms of the clinicians, they're not seeing those micro moments, so they're not able to understand the full picture and the full burden of disease, and therefore they could be missing or underestimating the disease. They could be having some errors in the estimation of the burden because they're not provided with the full picture, because it's quite difficult for people to remember all the little things that go on in their lives. And I think that's therefore it's quite an important thing to try and capture. And another one here is that it helps counteract the challenges of relying on memory. So I'm sure a lot of you are aware of recency bias and recall bias, but these are two things that kind of pop to mind. So it's quite well known that people remember the most recent event change that happened in their lives, and therefore this can be a bias to their memory. And likewise with recall bias. So it is quite difficult to remember something that was a while ago over a long period. And then again, I think of sleep. You know, if you were asked when you slept well and when you struggled, it might be quite difficult to then think of that over a long period of time. So what we'll do now is we'll move on to the case studies. So Mark, I'll pass over to you.
Thanks, Sally. So first case study is in a study we've been running for a while in Charcot-Marie tooth disease, very common rare disease. So this is an observational study across EU5 and the US. Got almost 3,000 patients that have been recruited into this study. It's going to run for 5 years. I think it might be year 4 at the moment. And very detailed data capture across a whole range of PRO instruments, but then the information about patient profile, demographics, in order to build up this pretty granular understanding of burden of disease. And the data capture, bring your own device, so people download a free smartphone app that they can use. Crucially, temporally proximate to these micro moments. And what we were hoping to do for this study, if you look at the next slide, is part of Charcot-Marie Tooth disease is falls. So patients have increasing problems with mobility and dexterity, and capturing falls when they only see their clinician, maybe once, the neurologist, for instance, once every 6 months, retrospectively, to understand how many falls they had in those 6 months, is quite tough. So it's a perfect example of what Sally was talking about. And it's not just falls, actually, it's another phenomenon is when they almost fell over. So we wanted to understand that and build up a big picture of that by this proximal data capture of these micro moments. So how we did that is we added this to the study app actually after it had launched. So we were able to do that quite a nice flexible way because our platform enables that. So we added a very simple counter that you can see in the screenshot here to the right of the screen, where a participant at the home page can simply press the button to record a fall, record an almost fall. And the value of that, as I said, is it encourages the patients to build those records. They can share those actually in this study themselves with their clinical team. So it helps them to build up a record of their history of falls that they can then take to their different healthcare professionals. And obviously for the study team, that was new data. The literature doesn't really have anything that granular about the impact of these falls in Charcot-Marie Tooth, so it's pretty exciting to get those data. Okay, next case study then. This is a case study in transfusion-dependent beta thalassemia, a study that it's completed now, but we ran a couple of years ago. If you go to the next slide, I'll give you a quick outline of what that the study design was. So this was not just in adults, but that was actually also in adolescents, 12 to 17-year-olds. International study, a couple of European countries in the US. And it was a relatively short, it was a longitudinal study, it was relatively short. And if you look in the, if you want more information about that, and the previous study actually as well, you see at the bottom, there's a link that you can click on in the chat to get a case study for this. It's quite a short study, 90 days, but ambitiously, we wanted, with the sponsors, Bluebird Bio, this was all being published, this is all publicly available information. We wanted daily data capture. And the reason we wanted daily data capture is that we wanted to understand the impact of transfusion. And we had the hypothesis that it was not only on the day of the transfusion itself, but we thought there might be an impact on the patient or on the caregiver on a day before the transfusion, 2 days before the transfusion, maybe a day or 2 days afterwards. And we would only really be able to get to that sort of granularity of data capture by this micro moment approach and getting daily data capture. So that's what we did. As you see, the importance of transfusion, they happen it varies a little bit, let's say about every 3 weeks, something like that for patients. Understanding the impact on days around the transfusion itself, and understanding the impacts, plural, because we thought there were a number of different ones. It could have been in terms of pain, fatigue, health-related quality of life, and things like also actually the administration time for treatments, particularly in the US actually, but for patients and their families. So you see a couple of the screen grabs for this study. And on the left, you see one of the surveys about fatigue we administered daily. And one of the challenges also with this ambitious data capture is getting people to do it every day. Okay, it's easy to try and ask for people to do it. How are we going to make sure that people engage with that? Well, for this study, we had a really nice reward program that we designed, which rewarded the various different actions that we were interested in people doing each day. And so, for instance, if you do a certain action, you get a certain number of stars, another action, you get a certain other number of stars, and you build up your number of stars. Then after about a month, you can cash those out for your Amazon voucher or whatever it might be, depending on the country. So that meant we have a really strong level of engagement over those for that daily data capture, enabled us to pick up the impact of the transfusions and the days surrounding them. And you know, those data were part of the way that we were able to inform the burden of disease of transfusion on this patient population prior to the introduction of new innovative therapy, which sought to reduce the burden of transfusions on the patients. So that's pretty much it for my two case studies. I'll pass you over to Sally.
So the next case study we're going to go through is one on capturing allergen exposure in peanut allergy. So I'll run through an overview of this. So this was an observational study with bring your own device data capture. The participants that we recruited into this were between 4 and 17 years old, with the younger age group, so the 4 to 11-year-olds were then completed by caregiver proxies, and then the older participants were then done by themselves. So then this was done in the US, and it was a 24-month data capture. So what we looked at here was details on demographics, the disease management, and several patient-reported outcome measures, which were then to quantify the burden of the allergy. So just a few, perfect, thank you. So I'm just going to run through the sort of the challenge, the solution, and the value here. So in terms of the challenge, it's always the way that these the exposure to these allergens are going to happen away from a clinical site, and therefore it's, like I was saying earlier, it's quite difficult to remember and not be subjected to recall or recency bias, and so that's in itself one of the main challenges. And then another thing is that actually what isn't often routinely collected is the data on who then isn't exposed to the peanut allergy, which is also really important to understand. So the solution here is we asked people to complete a weekly survey via an SMS message. So hopefully you can see the video on the right hand side. But what we did here is we asked participants on the weekly basis whether they had been exposed to the peanut allergy in the past 7 days. If it was a yes, they would just need to say Y for yes, and if a no, they would just say N. And I think the important part here is if they said no, they weren't given any more. They weren't asked any more questions, and they weren't given any more things to do. If they then said yes, so they typed Y, then they were then sent a link that they could then click, and it would go click into, and it would go straight to the survey questions, where they'd then be asked follow-up questions on that. So hopefully you saw that on the video on the right hand side. Yeah. So the value here is just that, I mean, mostly it's really useful data that we've collected with relative ease for the participant. The fact that there is also this collection of data that's then about the lack of exposure as well, I think that's not collected enough, but actually it's something that's then quite useful. And also importantly, that then only people have to complete more surveys, so limits the burden to them if they are exposed to the peanut allergy. And I think that's the key thing. They don't have to be burdensome by completing lots and lots of surveys if they actually didn't have this allergy. So I'll move on to the next. So this is the final case study. So this was a way to track symptoms in melanoma. So this was another observational study. It was in adult participants in the UK, another 24 months duration. Again, we looked at demographics, we collected information on medical profile, some monthly patient-reported outcome measures, and a symptom tracker, which I will go on to explain a bit more about in the next slide. So the challenge here, again, is that it's really difficult to remember how your pain's been, for example, for the last week, month, however long between the clinic visit. But actually, it's a really important symptom for participants with stage 3 and 4 melanoma. So it's something that is typically asked in a clinic visit and yet difficult for people to remember. So the solution here was to have a daily, like an e-diary, so a daily reporting of symptoms, which was then we were able to then share with clinicians, which just allowed for that more detailed information and capturing these micro moments. So as you can see on the right, that's just how the e-diary then looked and all the information we collected. So the value here is that it encourages actually more frequent interactions with the study app. And it really drives home the what's in it for me factor. So the participants are actually more engaged with this approach, and it is a lot about then the engagement for participants, and also, of course, that it's really important and valuable to clinicians if we can then send this information. They can then see, or this information can be shared with them, so they can get a bigger picture of the burden of disease. And this will also ultimately help with influencing treatment decisions and access to the most appropriate treatment. Okay. Back to you.
So thanks a lot, Sally. So I'm just going to sort of hopefully bring things together with a few strategies that we've seen across these different types of studies for capturing micromoments to help quantify these events. So to start off with, if you just want to click, that's the first one. Now, this may be obvious, but you know, patients are the experts on these micromoments, not you as a researcher. So we'll have some ideas about what those are, but we may not understand all of the range of the impacts that these things have. So get input from those patients. I mean, we found that in so doing, actually, sometimes our plans, our designs have changed quite radically, always for the better, I would say. And so get advice on how you capture those, but also the way that you capture them, making sure it's easy for participants. They will really give you fantastic feedback on that. So after that, as I said, the real, hopefully this has come out in the different case studies that we provided, but this has to be easy. If you have some very complicated requirement to capture a micromoment, you will lose the immediacy of that. So, you know, Sally's example with the SMS notifications is really nice. Everyone can get those very quickly, and as she showed, if you're capturing data on lack of an allergen, it takes seconds. So making it as easy as possible, particularly if you want very frequent data, is really important. So there's a part of that which is about UI UX type design, but similarly, even with all of that, really don't neglect doing user acceptance testing. It does take a bit of time, but frankly, in our relatively broad experience now, if you cut corners on that, you'll pay for it later, actually. So really do think about that. And then after that, pretty much all of these case studies we've talked about are about getting multiple data points from participants over time, so longitudinal. Now, it's very tempting to be over ambitious with your data capture for this. And in pretty much all the studies that we do, there's a prioritization session with the clients where we sort of think about all the things we could capture, and then we have to prioritize. It might be to say, well, don't do the second tier stuff immediately, or don't do it at all. But it's better to be a little bit disciplined about that and prioritize than to overburden and not get any data, which could be disastrous. And relatedly, I think rewards programs are absolutely critical. In all of the studies, and I think every time we've implemented a rewards program, they've resulted in pretty significant, I mean, it does vary depending on the circumstance, but a pretty significant uptick in terms of the engagement. So particularly when you're looking for lots of short engagement, working out a reward framework that is attractive for patients, and as Sally said, is part of the what's in it for me factor as to why they might want to engage in this data capture in that, very closely tied to their daily life. Rewards programs are really valuable for that. So they have to be tailored to the study and the cohort. But if you're doing a longitudinal study and you don't have a rewards program, I'd really challenge you to think about why not, and to challenge you to think about what is the benefits that a reward program could be. As I said, in our hands, they always have benefits. So I think that's about it. So those are our strategies. I think now we go to Anna for some Q&A.
Yes, Q&A time now. So yeah, if anyone fancies popping up some questions, we'll answer them. I'm going to dive into a couple I've got here already. So Mark or Sally, you jump on it if you want to answer it. So question number 1 is, should micro moments that are only important to the patient, so for example, not the sponsor, be collected? I don't know if Sally or Mark wants to answer that.
I'll try on that one. A few thoughts. One is that and actually, if you're doing a longitudinal study, I would be interested to know about what things are important to the patient that aren't important to the sponsor. Actually, I think if something's pretty important to the patient, sort of by definition, it should be important to the sponsor. Now, that doesn't necessarily mean that the function or the capture of that thing is going to contribute to the central sort of database upon which you will conduct analytics and research. As Sally showed, in the melanoma example, that was a feature that we developed for the patients, and we didn't actually use those data in our analysis. In that study, we were interested in the monthly PROs predominantly. So I would say that sponsors should always be interested in the things that are of interest to patients.
Great, thank you, Mark. And so we've got another question. Have you had any feedback from study participants about how burdensome they find reporting micro moments, such as via the SMS example that you showed earlier?
Do you want me to take it, Mark?
Sure.
Yeah, please add something if you need to, if you feel I've missed something. So for that particular example, that study's still still quite early days in that one. But we do have some wider initiatives that are going on to proactively then seek feedback on the different types and frequencies of data collection. But at the moment, we haven't done any satisfaction surveys, questionnaires with them to really understand this. But it is something that we will be looking into, and certainly something that we'll be talking about in our social media channels and blogs as well. I don't know, Mark, if you've got anything to add there.
Yeah, I'll just add a little bit of feedback from one of the other examples. So the falls tracker that we put together for Charcot-Marie tooth disease, that was something that the usability of that, if you like, was, we got some we didn't do a sort of standalone analysis on that. We got some we did ask patients how it was working, and so for that we've got a multi-stakeholder scientific advisory board which has patients from a range of the study countries, And they told us anecdotally on that they quite liked the way that was done. It was very easy, just essentially single click to record. They liked the way it built up the data, which was, you know, and we involved them in the design of that. So we were pretty happy. That obviously would have been very disappointed if they didn't like that, given that we involved patients in the design of that. So yeah, we've had some nice feedback on that. But I mean, I think there are some examples. It would be nice to, as Sally said, when we some of the studies are a little bit further advanced, to do something a little bit more systematic about the feedback of how these are used and viewed by patients.
Fabulous. I've got one more question, then I think we'll have to wrap it up, just as we're at time. It might be something to answer separately, but how can you implement these micro moments in discussions with the FDA and EMA, etc.?
So that's quite a broad question. I mean, I think these are the parts of data. This is just one example of data capture for these broader observational studies. And I would hope that if these are going to be used for discussion to the FDA and the EMA, prior to doing so, sponsors should engage with those bodies and ensure that they're going to be done in the most rigorous ways. So the ones that we've described here have not been for the purposes of submissions to FDA, but they have supported publications and contributed to HTA assessments. So obviously those regulatory bodies are different audiences. So all I can say is something relatively general about engage. If you are planning on using those, engage with those bodies to make sure you're doing it in the right way.
Brilliant. Okay, thanks, Mark, and thanks, Sally. We're just about at time, so we'll come back to some of the questions that come in through, but we'll come and answer those separately. So thank you for giving your names. I'll get back to you. Yes, and we look forward to seeing you all in next month's webinar as well, and hopefully see you at the conferences that are coming up, so ISPOR and ISOQOL as well. So have fun, everyone, and thank you so much, Mark and Sally. See you all again soon. Bye-bye.
Thanks all. Bye.